Dr. Aris stares into the glow of the monitor, his eyes tracking a complex, shimmering map of 13,609 proteins. For years, the human body remained a dark continent of biology, an uncharted territory of hidden signals and silent processes. But today, the silence is finally broken. The data-independent acquisition mass spectrometry results align perfectly. He clicks on the screen, and the digital atlas begins to rotate, revealing the hidden architecture of life itself. The journey from fetal tissue to healthy adult organs is no longer a mystery. It is a visible path, leading directly into the chaotic architecture of 25 different cancer types. This is no longer a collection of isolated charts or fragmented data points. It is a unified, spatially resolved landscape. Beside him, the lead analyst pulls up the t-SNE analysis, and the points on the graph begin to shift. They move into an orderly row, mapping the precise path of cellular differentiation with mathematical grace. Aris points to a cluster representing liver samples. It sits far from the fetal baseline, indicating an adaptive, plastic response to disease. The team watches in silence as the data breathes. They are seeing the body not as a static object, but as a dynamic system responding to the pressures of survival and decay. He moves to the next file, highlighting Module 8. The data shows a steady, progressive rise in proteins linked to the humoral immune response as tissues transition into tumors. Aris turns to his team, his voice low but filled with certainty. The brain remains stable, but the liver is reacting. We finally see the pattern. They pivot immediately to the practical application, drug repurposing. They cross-reference the map with drug bank targets, identifying potential on-target toxicities that explain why certain therapies fail while others succeed. Aris highlights the protein PLOD2, which flares up like a signal fire in 13 different cancer types. He marks it as a high-confidence target for future clinical trials. This is the breakthrough they have waited for. They continue filtering for plasma membrane proteins that could serve as anchors for antibody-drug conjugates. They find 41 candidates, including Tyrob, appearing consistently across 10 tumor varieties. Each click of the mouse is a step toward a cure. Each data point is a life that might be saved. The complexity of the proteome is finally being tamed, turned from a chaotic storm into a navigable map. They are no longer guessing. They are calculating. The landscape of oncology is shifting beneath their fingers. As the clock ticks, they integrate CRISPR essentiality data, confirming that MET inhibitors could be lethal to colorectal carcinoma cells. The evidence is undeniable. Aris saves the final dataset, closing the window on a decade of biological guesswork. The uncertainty that once defined their field has been replaced by the cold, hard clarity of molecular evidence. The simulation is complete, and the results are definitive. The map is complete. By mapping the proteome's spatial distribution, they have built a digital navigator for the human body, turning raw molecular data into a precise roadmap for the next generation of cancer therapies. The future of medicine has a new coordinate system,